The parts of a neuron. Dendrites receive messages from other cells. The cell body contains the nucleus, mitochondria, and other organelles found in other normal cells. The axon transmits signals from the cell body to other cells, and the synapse is the gap between the synaptic terminal and dendrites, where neurotransmitters are released and a signal is passed to other cells. Action potential. Ion channels allow sodium-potassium ions to diffuse across the cell membrane, and sodium-potassium pumps actively transport sodium-potassium ions across the cell membrane. Sodium ions move into the cell, depolarizing the membrane and creating an action potential. The depolarization causes sodium channels to open, depolarizing other regions of the membrane and moving the signal down the axon. Potassium channels open, potassium ions flow outward, repolarizing the membrane. This wave of depolarization-repolarization propagates through the axon, causing the release of neurotransmitters at the synapse. Graph of membrane potential. At section 1, old channels are closed and no action potential is present. At section 2, a stimulus opens sodium channels, and if the depolarization reaches the threshold, an action potential is created. At section 3, depolarization opens more sodium channels, making the inside of the cell positive as the sodium ions rush into the cell. At section 4, sodium channels close and potassium channels begin to open. At section 5, potassium channels close and sodium channels open, repolarizing the cell. After repolarization, the cell is back in its resting state. Neurotransmitter release. An action potential arrives, depolarizing the presynaptic membrane. The depolarization opens voltage-gated ion channels, triggering an influx of calcium ions. The elevated calcium ion concentration causes synaptic vesicles to fuse with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. The neurotransmitters bind to ligand-gated ion channels in the postsynaptic membrane. This allows potassium and sodium ions to diffuse through. Sodium channel blockers. These toxins prevent sodium ions from flowing in and out of cells, preventing the formation of an action potential and the repolarization of a membrane after an action potential has been created, as can be seen in part 4 of the membrane potential graph. Both of these outcomes prevent the neuron from passing a signal onto other cells.